Welcome to the Chapter 7, Section 2 podcast. This is on eukaryotic cell structure. And we're going to be looking at all the organelles in a cell. So let's start with this. So the first thing we're going to talk about is the structures that are within a eukaryotic cell that perform important cellular functions. They're known as organelles. And we're going to divide the cell into two major parts: the nucleus and the cytoplasm. We're going to talk about what's in the nucleus and what's in the cytoplasm. The cytoplasm is the portion of the cell outside the nucleus. If this is the nucleus here, then the cytoplasm is going to be all those parts outside the nucleus. Here's a picture of a plant cell, and we talked about this in class, and you can see these are all the organelles that we're going to refer to in this class. And just two things I want to show you. Notice that there's a cell wall here. You're not going to see that in the animal cell. The cell itself is a little squarish, and you can have a large vacuole here. And the other big difference is going to be the chloroplast. So these are the big three differences you're going to see in a plant cell. Here's an animal cell, and you can see there is no cell wall. It's more of an irregular shaped. There is no cytoplasm in it, and those in the animal cells more about what's missing than what there is there. So what's the function of the nucleus? It's a control center of the cell. You can see I put a little control center, a NASA control center, and it contains nearly all the DNA, cell's DNA. There is some in the mitochondria, and these are DNA is the coded instructions for making proteins and other important molecules. Here's the nucleus itself. You can see there's a nucleolus in the center of it. There's chromatin all throughout the inside of the nucleus, and then you have a membrane here with the nuclear envelope, which is the membrane, and then you have these nuclear pores with these holes. And what's the purpose of the nuclear membrane? The nuclear membrane is nuclear envelope is two membranes, and just basically keeps things in and keeps things out, and the nuclear pores allow material to move in and out of the nucleus. That's once again these little holes here. So the granular material is chromatin, and chromatin is the DNA bound to proteins, and we'll talk more about that when we get to DNA and RNA. DNA, when it condenses, you'll see it as chromosomes. So now you know what the chromatin is. It condenses into what we know as chromosomes, and chromosomes are what have the genetic information that you pass on to the next generation. Inside you also have a nucleolus, and this is where assembly of the ribosomes begin. What's the function of ribosomes, by the way? And the ribosomes is all about making proteins. Proteins are assembled on the ribosomes, and the ribosomes are made of small particles of RNA and protein, and they're found all throughout the cytoplasm. You don't just find them in one place, find them throughout the cytoplasm. And here's a look of it of a ribosome. You can see the ribosome contains these proteins here, and then there is this tRNA here that looks at the information. Here is a messenger RNA, which is not part of the ribosome. It comes from the nucleus. And here's the protein itself made of the amino acids. This is not part of the ribosome also either. So the ribosome really is this the blue ribosome here in your transfer RNA. Well, transfer RNA is not part of the ribosome, but it's in conjunction with the ribosome. So cells are active in protein synthesis are packed with ribosome. That makes sense. The endoplasmic reticulum, and this is an internal membrane, meaning it's membranes inside the cell, and we call it the endoplasmic reticulum, or ER, and it's where lipid components of the cell membrane are assembled, and there are proteins assembled there if there are ribosomes, and other materials that are exported from the cell. This is where things are made for the cell membrane or for export. And here's your ribosomes on the endoplasmic reticulum, and there are two types of endoplasmic reticulum, or ER, rough and smooth. And the ones that have to do with protein synthesis are called rough endoplasmic reticulum, or rough ER. And the reason why they're rough is they appear to be rough because there are ribosomes on the surface of rough ER. And rough ER you're going to find in cells that produce large amounts of protein for export. Smooth ER does not have ribosomes on its surface, so it has a smooth look to it. And these do tasks such as synthesizing membrane lipids and the detoxification of drugs. They have different enzymes that do specialized tasks. The Golgi apparatus. And the Golgi apparatus is sort of like a place where the proteins are moved to after they're made on the rough ER. And they look as stacks of membranes. And the Golgi apparatus modifies, sorts, and packages proteins and other materials that and they're stored in the cell, or they secrete it outside the cell. The big word here, remember, is things are shipped to their final destinations from the Golgi apparatus. Lysosomes. Lysosomes are organelles filled with enzymes, and they break down parts of the cell, such as lipids, carbohydrates, proteins, and other molecules they can use for the cell. And they also break down old organelles. So they're kind of a cleanup crew in many ways. What's the function of vacuoles? These vacuoles store materials such as water, salts, proteins, carbohydrates. You can see this big space right here is vacuole, and this is a plant cell. You can see there are chloroplasts and cell wall, and if you look at some plant cells, you can tell where the vacuole is just because there's nothing else there and everything's crowded around the vacuole. In plant cells, you're going to find one single large central vacuole, and this is useful because the pressure of it allows plants to support heavy structures such as leaves and flowers. And you can see here's a vacuole here, and you can see there's nothing else there. It's a large vacuole, and it gives the cell some structure. And you also find vacuoles in other unicellular organisms, and for instance, such as paramecium, it contains a contractile vacuole that helps it pump water out. Now the mitochondria are found in all eukaryotic cells nearly, and what they do is they convert chemical energy stored in food into compounds that the cell can use. Basically, they convert food into the energy that the cell can use. And there are two membranes within a mitochondria. You can see these folds here. The inner membrane is folded inside, so it has this characteristic stripe kind of look to it. And if you look back at this picture right here, you can see the membranes inside the mitochondria there. And so the inner membrane is folded up inside the cell. And that's to increase the surface area for reactions to occur. What is the function of chloroplasts? Chloroplasts are found in plants, and they capture energy from sunlight and convert it to chemical energy, and that process we know is photosynthesis. And they're chloroplasts, they're green. They contain the green pigment chlorophyll, and we'll study more about that when we talk about photosynthesis. What are functions of the cytoskeleton? The cytoskeleton give shape and internal organization to the cell. And you can see that yellow line showing you the cytoskeleton, and it's a network of protein filaments. And it also, the two big things is it helps the cell maintain shape, and it's also involved in movement in some cases. So there are two parts to it, the microfilaments and the microtubules. And here's a look at it. Here's your cell membrane, and here is, you can see some organelles around it, and here's your microfilament here, this green guy, and here's a microtubule, this blue tube right here. So the microfilaments are, they're kind of thread-like, and they produce a tough, flexible framework that supports the cell. And in some cases they help the cells move, and we'll talk more about that later on. And microtubules are hollow, and they also maintain the cell shape. You use them in cell division. We'll talk about this when we talk about mitosis. And they also allow cilia and flagella, and these are things that help the cells, some cells swim, and that that's part of the movement part that we talked about. So we have centrioles, and the centrioles are used in cell division. Here's a centriole here, here's a centriole there, and they're found near the nucleus. Now let's look at a comparison here. Now what we're going to look at this first chart is prokaryotes and eukaryotes together, and what do they have in common? They both have a cell membrane, ribosomes, and a cell wall. The eukaryotes have these organelles here, and let me go back and make and you can see the prokaryotes don't have any other organelles except for these organelles right here. Animal cells, the plant cells have a cell wall and a chloroplast. The animal cells have lysosomes. Remember, those are the packed with enzymes that I talked about that break things down. And then these are all the organelles that they share in common. On to the quiz.